Hi, my name is Alexander Barang. I am live here in Vienna from at TissueGnostics. I'm actually in home office, but even still, I'm live, and I'm here with Dr. Kim Blenman, who is an assistant professor of medical oncology at Yale University in the Department of Internal Medicine and Yale Cancer Center, with a secondary appointment in computer science. She uses and develops novel software tools to understand the mechanisms responsible for disparities in disease pathogenesis and therapeutic responses in humans. And so without further ado, I will let Kim Blenman have the floor.
Fantastic. Good morning, everyone. First and foremost, I'd like to thank TissueGnostics for allowing me this time to share my work with you. The topic that I will discuss today is the spatial dynamics of the human tumor immune microenvironment, or TIME. So there are three key concepts that I hope you take away from this talk. The first is the use of an application of imaging analysis tools in clinical research. The second is illustration of workflow for tissue cytometry utilized in interrogation of time with visual verification of cell and tissue compartment isolation and identification. And the third is characterization of spatial dynamics of time components through machine learning and deep neural networks for high content phenotyping. So I have a multi-layer approach to this process. The first layer involves multiplex tissue staining. And normally I would do this either by immunohistochemistry or by immunofluorescence. If I am doing between 7 and 8, looking at between 7 and 8 targets, I would use immunohistochemistry. Anything above that, I would use immunofluorescence. So this is an example of the 7-color immunohistochemistry, and this is an example of the standard immunofluorescence. So the second step to this approach is actually whole site imaging, which requires high resolution with automated scanning. And for instances where I am using the immunofluorescence microscopy, I actually rescan the same image or same region with saved region gates for in silico analysis in order to fully interrogate multiple, more than 8 targets at the same time. And then the third layer of this approach is the image analysis itself. And that's actually going to be the main focus of this talk. So the image analysis that we do is focused on identifying each cell by immunophenotyping using in silico methods for the multiplex imaging and the tissue cytometry. And what I'll be talking about mostly for this session is the latter two. So looking at correlating changes with clinical parameters and outcomes and therapeutic response, but mostly for this talk about assessment of spatial relationships. And for this, actually, we use the TissueGnostics TissueFAXS Spectra system and software. And as you can see quite nicely, as an example of the software itself and some of the clinical analysis outputs that we do get. And I just like to just very briefly point out that the workflow for the immunofluorescent microscopy has been previously described in detail in this paper in Cytometry A. And the workflow itself is detailed here. And I'll just touch on it just a little bit to just orient everyone with where we are going with this. So the workflow is for this, in this case, is for histology and imaging analysis for in silico multiplex images, but you can use this for just single step analysis as well. So the first step in this process is to literally just do the multiplex histology. However you whatever is required for your multiplex sample analysis. The second step is to do image acquisition. And in our case, we actually do whole size scanning. And then if we are doing the antibody conjugation with two targeted fluorochromes such as Cy3 and Cy5, we'll do a dye inactivation process so that we can do multiple rounds of this staining using, as I mentioned before, at least 8 target antigens of interest. And so that's part of our step 3 process as we go back and forth. And once you inactivate the dye, you will do an image acquisition and then go back to the staining again. So once this step is finished, we will go on to image preparation, which requires image stitching performed through reconstruction of the whole image from tiles from each staining round. And then we will move on to a process of in silico image creation, whereby we take each of the individual images and overlay them on top of each other based on the registration that we have identified through DAPI channel staining. And so this is actually a very nice step that allows you to actually go back even, days or weeks after you've stained a slide, uncover, slip that slide, and restain it with something else new. And so it allows you to keep that exact same orientation and structure of that image so that you can combine different antigens, mix and match different antigens to address various questions that you have for your tissue microenvironment. And so the most of our talk that we will discuss, most of the presentation that will be discussed today will actually be on steps 6 through 7, which focuses on the cell phenotyping, the tissue cytometry, and the visualization techniques. And using those 3 constructs to really define and outline some of the methods and approaches that we can use for spatial relationships, for identifying spatial relationships. And so here is a very nice picture of the TissueFAXS Spectra system that contains the 2 monitors, the camera, as well as the stage that houses at least about 8 tissue sections. And so what I will be discussing today is breast cancer, and that will be the example that I use throughout this presentation. And with breast cancer, I will show you that an example of the immune microenvironment of triple negative breast cancer. And the reason why I'm using triple negative breast cancer as a model is because it is the subtype of breast cancer that has high, as a hallmark, has high infiltrating tumor immune cells. And these infiltrating immune cells that you can see quite nicely on hematoxin and eosin stains, you can see quite nicely as we use different biomarkers for each of the different cell types. You can see how complex that system is. So for example, our CD8 T cells are highlighted in yellow. CD4 helper T cells are highlighted in red. The CD20 B cells, a beautiful example here, are highlighted in orange. And our cancer cells, our breast cancer cells, which are tagged with pan-cytokeratin, are highlighted here in green. And then in blue, we use a marker called DAPI as a marker of all cell types. So whether or not, regardless of whether they are stained with an actual antigen. So although we see this immune microenvironment, we see how complex it is. And that complex structure is really a mixture, an abstract mixture of shapes, colors, and textures. And those shapes, colors, and textures actually need to be separated in order for us to identify individual cells in order for us to identify individual compartments, if we're interested in looking at, for example, the tumor bed versus the stroma, or if we're interested in looking at specific structures within the tumor microenvironment. And so therefore, we need ways to actually tease these different pieces and parts out in order for us to do the spatial analysis that we need. So because the spatial analysis itself actually requires that the isolation of those components, as well as linking of Cartesian coordinates to those components so that we can do the types of distant measures that we need for the spatial analysis. And so a very simplistic workflow for the spatial dynamics is to start out with, the simple spectral unmixing. And then from the spectral unmixing to separate out the different cells into different channels so that we can identify the individual cells. We can also do tissue segmentation. Okay, in this case, we will be interested in separating the tumor bed from the actual stromal components. And then with that, we also loop back and we will do nuclear segmentation, because that nuclei segmentation is actually required in order for us to not only isolate those individual cell populations that I showed you, but also to identify the specific cell populations. And then as part, the majority of this talk is actually going to be on the latter half here, which is actually showing examples of distance and proximity mapping, as well as cell connectivity mapping. And so the distance mapping is literally looking at the distance of the immune cells from the tumor bed. And then the proximity mapping is actually a really nice tool that we are exploring to really identify very specific regions within the tumor microenvironment and look at the components within those regions and their relationship to, for example, the tumor bed, or maybe a blood vessel or a lymphatic vessel. And then finally, we will talk about, a nice tool that really explores cell connectivity mapping. And what that is that within our tumor microenvironment, it's split into various pieces. And those pieces, for example, may shed some light on how the dynamics of the different immune cells are interplaying with the tumor cells themselves. So if we can, for example, gain some knowledge from understanding how many cells are, how close cancer cells are to specific immune cells. So for example, we use a cutoff of 50 microliters for this data that I'm showing you, that I will show you. It may give us clues as to maybe how different, in our case, therapeutic responses are playing out based on, the connections and their interactions. And so the cell connectivity itself is actually going to be very important. So although for what I'm showing you in this presentation will be restricted to about 50 microliter distances, we actually plan to actually do a few further studies where we're actually looking at cell-cell contacts, because that's going to be important as we look for how these different cell types within the microenvironment are interacting. And so the first thing I'll talk about is the spectral unmixing. And so just very briefly, this is an old process, and very just in a nutshell, it really focuses on having a unique fingerprint, for example, each fluorochrome that you have attached to the biomarkers of interest. And each of these unique fluorochromes, as you can see here, the example here in green, has a profile, a spectral profile that is different, as you can see here as I trace here for the green, than the other profiles. And this profile is characterized from 400 nanometers all the way up to 700 nanometers. And then from that, you can piece these out into very nice categories that allow you to do something a little bit more complex, as I show here with unmixing, in this case, of 11 different biomarkers here. And each of these biomarkers I can show you on the x-axis is the biomarker of interest, or the fluorochrome of interest, and then on the y-axis, and then on the x-axis is actually the wavelength. And you can see for each of the fluorochromes, you have very unique profiles that you can help to identify each of these different fluorochromes. And so what's the advantage of spectral unmixing? So on the left-hand side, this is literally the raw image. And as you can see in the raw image, we have a lot of background. So you have things like the background fluorescence that you get from the tissue itself, as well as the background fluorescence that you get from these pieces, such as the red cells that you see here, right? And so the advantage of being able to do a spectral unmixing is that is what you see on the right side, whereas you get rid of all this extra noise and you help to enhance your signal, okay, so that the biomarkers that you're going to be analyzing can be expressed at a higher threshold, and your signal to noise allows you to actually do better in terms of your segmentation. So that includes not only the tissue segmentation to identify compartments, but that's also your cell segmentation to identify the specific phenotypes and specific cells that you see here. And so this is where I get to the next step, which is actually the deep learning nuclear segmentation, which is the other advantage of being able to do the spectral unmixing. So you see here in the normal segmentation, we do have, these clusters that tend to form, especially when cells are very close together. When we use our deep learning tool, you can see that you get a better resolution for the nuclear segmentation, and this clustering tends to resolve quite nicely. So now I'll focus on the actual spatial relationships and give you a very nice example of how we use these tools. And so we have recently published a clinical trial where we used an anti-PD-L1 immunotherapy plus standard of care chemotherapy in 50 triple-negative breast cancer patients. And as I mentioned in the very beginning, these triple-negative breast cancer patients, the hallmark of these patients is that they actually have very high levels of TILs, comparatively speaking, with their counterpart subtypes. And as you can see in this particular graph where I show the percentage of TILs in patients who have a pathologic complete response, so that meaning that these patients actually have no cancer after we treated them, or the patients who have residual disease, or RD, meaning that patients still have some level of cancer after we treated them. And you can see that in the patients with the residual disease, they actually have significantly lower tumor infiltrating lymphocytes. And this is actually not only important, but because of that, we need to understand what is the cell makeup in these residual disease patients, as well as the cell makeup in the patients who actually do a lot better, so have no cancer cells left. And so to do that, we using the tools from this TissueGnostics platform to really interrogate the tumor microenvironment. So the key questions that we have is, what is the distribution of the key immune cell populations? And what are the spatial relationships between the immune cells and the cancer cells? And so for the last question, we have 3 key measurements that we're going to look at. The one is the proportion of the immune cells within the tumor microenvironment. And 2 is the proximity of individual immune cells to the tumor bed. And then 3, the number of cancer cells within 50 microns of a specific immune cell. And so the 5 specimens that we're using is that we have 25 patients with PCR, meaning that they have no cancer cells after treatment, and 25 patients with RD, meaning that they have residual disease, they still have cancer after treatment. But the biospecimens that so these are the patients that we're using, but the biospecimens that we're using are actually going to be the samples, so pre-treatment, formalin-fixed, paraffin-embedded core needle biopsies. So meaning that these are going to be the samples that we collect before the patient's actually treated. And then we're going to take those samples and we're going to stain them for these various biomarkers. So for B cells, we'll use CD20. For cytotoxic T cells, CD8. For helper T cells, CD4. For breast cancer cells, pan-cytokeratin. And again, as a marker for all the nuclei, we use DAPI. So we start out with these two processes, so the spectral unmixing and the tissue segmentation. And I will just show you the spectral unmixing because we've already discussed this, so I will not go into details of that. But what I will talk about is the tissue segmentation. So as I mentioned, the tumor microenvironment consists of two major compartments. One that I show here is the tumor bed that you see here highlighted in green, and the stroma, which contains the the immune cells, as well as fibroblasts and other stromal components. And so these are the key components that interact to resolve the tumor. And so the first thing that we need to do is actually to segment this tissue so that we can really separate, so identify and isolate and separate the background from the stroma and the actual tumor bed. And so here is the actual sample itself. And so the stroma is this segment here that you can see is relatively in pink. The tumor is this segment here that's in green, okay? And the background is this sample, this area in black. So when we segment these areas, areas as shown in segmentation are highlighted actually and extracted as a white mass. So this white area here is actually the background. The white area here in this panel is actually the stroma. Okay, so these are the region that contains the immune cells. And the white area in this panel is actually the tumor. And what we do is that we also reconfigure this image so that it combines both the stroma and the tumor so that we can really isolate each one of these compartments and look at distant metrics between the two compartments. And then we do nuclear segmentation, which I will not go into, but again, you saw in the previous figure, where quite nicely you have the contour mass showing the nucleus of the tumor cells. And then you can see quite nicely in the stroma, the nucleus of the immune cells are also highlighted. So our first question is, what is the proportion of CD4 or CD8, CD20, and cells or the general immune cells in the cohort? And using the process that I've just described, here's the distribution within our population. Again, in our 25 patients of PCR and our 25 patients with residual disease, we see that our most abundant immune cell population is going to be the CD8 population. And then after that, we have the CD4 population, which is in gray. And in the blue, the dark blue, is the B cell population. And this actually makes sense because of their role. So our CD8 population are going to be the population of cells that need that cell-cell contact to kill the cancer cells. Our CD4 population are going to be the cancer T cells that help the B cells and the CD8 T cells by interacting with them and activating them, and also giving them cytokines. And our B cell population is going to be the lowest amount because their role is actually to not only secrete cytokines, but their major role is to secrete antibodies. So the amount of cells needed there are not going to be as much. And so now I'll talk a little bit about how we use the distance and proximity mapping tools. The first example I would talk about is going to be the distance tool. And so the key components of this is visualization of the distance distribution using actual scatter plots. So the question that we had for this is, what is the immune cell distribution by distance from the tumor bed? And here I show an example field of view, just one field of view, where we show the CD20 positive cells, which are in orange, in relation to our tumor bed here, which is in green on all three sides, right? And in order to better visualize that, we constructed a scatter plot with the individual showing individual B cells as individual dots. On the x-axis are the cell sizes of the B cells, and on the y-axis is the distance to the tumor bed. We separated this graph into 7 different regions that represented ranges of distances from the tumor bed. So ranging from 0 to 25 microns, from greater than 200 microns. On the right-hand side, I show a plot of the actual data. So this is the tumor cells. These very beautiful, nice, large cells are the tumor cells. Here in the middle is the actual stromal component. The cells in all these graphs, the cells that are positive for the attribute of interest, and the attribute of interest is going to be here highlighted in top in blue. So the attribute is a range from 0 to 25 microns from the actual tumor bed. Those cells that are positive will be highlighted in red. And those cells that are negative, meaning that they are not within 25 microns, 0 to 25 microns, are the ones that are going to be highlighted in blue. And so this gives you a very nice picture of the distribution of those cells. And next here is the distribution from 25, just again focusing on the red cells, the distribution of 25 to 50 microns, the distribution from 50 to 75, 75 to 100 microns, 100 to 150 microns, 150 to 200 microns, and 200 plus microns. And again, this is just one field of view, so you're not seeing the full range of the rest of the tissue. But when we put all this data together, and we actually look at the distribution through this distance mapping, it gives us actually a very nice profile. So on the x-axis, we show individual patients. And on the y-axis, this is the percentage of CD20 positive cells. The rows represent the ranges from 0 to 25 up to the last row, which is 150 to 200 microns. As you can see in both the PCR, so the patients who actually had no residual disease and the patients who actually had residual disease, the highest percentage of immune cells are the ones that were in within 25 microns of the actual tumor bed. And what's interesting is that if you compare the PCR with the RD, we can very nicely see that we have a lot fewer B cells in the patients with residual disease compared to those that actually had a very nice response to the cancer treatment. So next, I present CD8 cytotoxic T cells. So these are the cells that are in yellow. And again, CD8 T cell size is on the x-axis. The distance to tumor bed is on the y-axis. Each dot within the scatter plot represents a unique immune cell. And we look at the different ranges. from 0 to 25 microns to 200 plus microns. So in red, we see the samples that are the cells that are within 0 to 25 microns of the actual tumor bed. In red, we see the ones that are between 25 to 50 microns. As well as those that are within 50 to 75 microns. And you see very few as we get further up between 75 and 100 microns. And now between 100 and 150 microns, 150 to 200, and 200 microns. Now, when we put all of this together with all of our patients, we once again see with our individual patients on the x-axis, the percentage of CD8 positive T cells on the y-axis, and the ranges in the rows. We can see for both the patients with PCR and those with residual disease, we can see that, again, most of our cells are within 25 microns of the tumor bed. And then also, once again, the number of CD8 positive cells is actually fewer in the residual disease patients than in those who actually had a very nice response with the PCR. And then finally, we talk about our helper T cells. So these are the cells in magenta. Again, same graph. In the red, from 0 to 25 microns, we see very nicely our cells that are positive. From 25 to 50 microns, from 50 to 75 microns, from 75 to 100 microns, from 100 to 150, 150 to 200, and from 200 plus. So as we look at the distant map, again, we see the same phenotype where most of our cells are within 0 to 25 microns. So the interesting feature about our CD4 helper T cells is that there is actually no difference between the distribution for patients with residual disease and patients with PCR. So what are our findings from this distant mapping? So the majority of cells from all the immune cell populations are within 25 microns of the tumor bed. There are fewer B cells and cytotoxic T cells in patients with residual disease versus those patients who had a complete response, meaning that they had no cancer cells. And the most prominent effect was seen in the B cells. And interestingly, there was no difference in the helper T cell distributions between residual disease and PCR. So the other way that we actually have decided to use these tools is to use them as a way to measure the proximity to various regions, areas, or groups. And as one can imagine, this is very useful if you're wanting to look at, for example, the distances from an actual structure, such as a lymphatic vessel or a tertiary lymphoid structure or something of that nature. Or if you just want to, you have specific regions that you want to focus on and just collect data within those regions. So it may be a hot spot with a specific morphology of cancer cell that you're looking at. a specific architecture of the cancer and the immune cells that are of interest. And so these are the specific sort of questions that you can sort of like address with using this proximity region tool. And the way that we decided to use this is to actually build a map within the actual stromal region. And as I mentioned before, the stromal region contains our immune cells. And if we can break those immune cells into segmented areas from one, and actually this is from literally 8 different regions. I'm only showing 5 regions here because of the actual size of the image. But here you can see that you can very nicely segment these out into regions, into areas, and you can measure multiple things, such as the amount of, in our case, immune cells within these regions, or you can measure the distance from the tumor bed to the actual different areas of interest, especially if those areas of interest have a unique distribution and makeup of the immune cells of interest. So this gives you a good way to really group things together versus what I previously showed with the distance metric, where I was looking at individual immune cells. Here I'm actually grouping things together and looking at distributions of groups. So that's an opportunity to address a lot of different nuanced questions using sort of that approach. But in what I'm going to show you now is more about us using this as a method to look at the distribution of immune cells within these specific areas. So in this context, we first talk about B cells, and the B cells, the size, so each dot again represents a single B cell, and that size, as you see, is displayed here on the x-axis, and the segmented area. So as I mentioned, we have actually 8 areas that, and I only showed you 5 on the previous image, that we have segmented. And one of those areas include the area inside the tumor region. And what I will only show for this is actually the area inside the tumor region and the area 1 region. And so as I show here, this is the area inside the tumor. And you can see in red where the actual tumor bed, the actual CD20 positive cells or the B cells are located within each of these tumor beds. So these are the areas in red. And then here is the area 1, which is near to the edge of the tumor bed itself for the B cells. And for the cytotoxic T cells, so this is the CD8 T cells, you can see in the tumor bed how it's a little bit more invasive. And then here is the region just around the border of the tumor bed. And then for the helper T cell, again, very invasive in some of these areas. But then for the proximity or the region around the tumor bed, you can see very few here. And so I only show a few of the results because the results are actually really similar to the distant mapping that I showed. But what this actually highlighted for us was that, one of the things that we tend to forget is that we spend so much time looking at the stromal components or the stromal region, because it has, it's going to contain the bulk of your immune cells, that we actually forget that the actual tumor bed itself actually does contain, immune cells, and those immune cells within the tumor may either be, agents for the tumor or agents for the immune system. And so actually paying a little bit more attention to those and trying to phenotype and map those out is actually would be helpful to try to delineate some of the therapeutic responses, potentially the therapeutic differences in therapeutic responses that we're seeing. And so finally, the final piece that I'd like to talk about is this concept of the cell connectivity mapping. And to give you just a little bit of background on why, we were interested in this is that, as you may know, for all of our cells within either any of the microenvironments, whether it's tumor or just normal cell microenvironment, especially for the immune cells, the way that they talk is through cell-cell contact and through close interactions. So if we can somehow identify specific patterns that are associated with, these spatial distances, it may help us to better identify what's going on within the tumor microenvironments. So as I said, so even for in order, for example, the CD8 T cells to actually kill the tumor cells, they actually have to interact with them, and they have to actually be within a certain distance of the tumor cells in order to have those effects. So that's why, looking at especially the cells that are within the tumor bed is also of interest, because those are the ones that made it in and are able to potentially be able to affect change and to kill those cells within that tumor within the tumor bed itself. Those that are within the stroma piece are the ones that didn't make it in, so aren't able to get in as they really need to. And so if we can figure out why that is by, phenotyping those and see if there's any links to the what those actually cells are doing based on their distances, who they're interacting with based on their distances and based on their interactions, we may be able to pull some more information out of what's going on in that environment at the time that we're assessing them. And so with that being said, what is the number of cancer cells within 50 microns of a specific immune cell? So this was based on the fact that, our tumor cells tend to be quite large, so between the 30 to 50 micron in size. And so we chose that as a good marker for something that's going to be quote unquote near, so to speak. And so this is where we use our connections, or our connectivity map. And as you can see with this particular graph, I show you quite nicely here an example, just a representative example of our B cells that are within the stroma, and the lines that are connecting from the B cells in orange to the nearest cancer cell that's within literally 50 micron distance. And so in this case, this really helps us to see what's connected, or who's in whose sphere of influence, is what I like to say. And to help visualize that, we use again a scatter plot, dot plot, as well as an actual visual image. And so on the left-hand side, we show a scatter plot with, again, each individual dot represents an individual cell. On the x-axis is the immune cell. In this case, this is the CD20. And on the y-axis is the number of connections to cancer cells. Now, because we realized from our first analysis that we had very few cells that are going to be in this region, we split these cells into groups, and to 4 groups to be specific. The first group is cells that have literally no connections. The second group are cells that have 1 connection with a cancer cell. The second group are cells that have between 2 to 3 connections. And then the fourth group are cells that have at least 4 connections, has at least connections with 4 cancer cells. And so on the right-hand side, I show you a nice image of that. So this is a cell, an image of B cells that have 0 connections. So the ones that have 0 connections are the ones that are in red, and you can quite nicely see that that's indeed the case. There are no lines going towards any of the immune cells. I'm sorry, any of the cancer cells. This is an image of cells with at least one cell. So here's a nice sample in red, and our cancer cell here, showing a line that is going towards that. This is one with at least two cancer cells in connection. So here's a very nice example of one here in red with one and then two cancer cells. And you can see this throughout this image. And here's a nice example of 4+, right? And so with the 4+, again, we're looking at the guys that are in red. So here's a nice one here showing an example with the 4+. And so what does this look like for our entire cohort? So as we look for these B cells, we see that the majority of our cells, regardless of whether these patients had a very nice response to therapy or whether they had residual disease, we see that the majority of our cells are not within the 50 micron distance of a cancer cell. But those that are, especially for the samples with the PCR, we see that at least there are at least 2 to 3, which is shown here in gray, cancer cells that are linked to at least one immune cell within most of these cases and most of these patients. When we look at residual disease, we see that this is not the case. And as a matter of fact, we have very few, most of our cases have less than 2 connections with tumor cells for these B cells. And so very few. And so next we do the same thing with the CD8. And we can see that within the CD8, again, with the CD8 cells being on the x-axis and the number of connections to cancer cells being on the y-axis. When we have no connections, you can see all these cells in red with no lines going towards any of the tumor cells. When we increase the connections to 1, we can see quite nicely the cells that have at least have 1 connection to a tumor cell. And then we can see quite nicely here cells that have connections to 2 to 3. So here it's 1, 2 cells from this particular cancer cell. And here's 4+. Again, 4+. So when we look at the side of these cytotoxic T cells, again, most of our cells have no cancer cells connection. And those that we have a reasonable distribution of CD8 T cells for all of our patients with PCR. But we have a lower frequency of those cells for our patients with residual disease. When we look at our CD4 population, so our helper T cells, again, here are the connections in red for those with no connections. And we can see at least 1 connection for those in red. Here is an example. And those with 2 to 3 connections. Here is an example. And then as we see those with 4 connections, we see actually quite nicely here is a great example. And as we take all of our sample, our information, and put it together, we see again the same phenotype, where the majority of our samples are not within 50 microns of a cancer cell. And unlike the B cell and the CD8 cytotoxic T cells, the distribution for the CD4 cells for the PCR and residual disease is roughly the same. So what are our findings here? So the majority of immune cells had no connections with cancer cells within 50 microns. B cells and cytotoxic T cells have fewer connections in patients with residual disease. compared to those with PCR. So of those connections to the cancer cells, fewer immune cells had more than one connection for those patients with our residual disease. And connections between helper T cells were similar between the patients with PCR and residual disease. So in summary, what have we learned overall? So in pretreatment triple-negative breast cancer tumor microenvironment, we see that the distribution of the immune cells, we have more cytotoxic T cells than we do helper T cells and B cells, which is again, confirmation of the function. When we look at the spatial relationship for proximity of individual immune cells in the tumor bed, we find that the majority of cells for all immune cell populations are within 25 microns of the tumor bed. Fewer B cells and cytotoxic T cells in the residual disease patients than those that achieve PCR are seen, most prominently in the individuals in the B cell subset. There's no difference in helper T cell distribution between residual disease and PCR. And it also reminds us from the data that there are actually appreciable number of immune cells in the tumor bed, and this is something that we should follow up on in additional studies. And that there are a number, when we look for spatial relationships in terms of number, the number of cancer cells within 50 microns of specific immune cells, we see that the majority of immune cells had no connections with cancer cells within 50 microns, and that B cells and cytotoxic T cells have fewer connections, meaning less than 2, in patients with residual disease versus PCR. So again, this is a very basic flow. And this actually opens up these new tools that are presented for the spatial dynamics, opens up the possibilities for a lot of new questions and a lot of new directions for not only the clinical trial space, but also for other clinical research. And these are my collaborators at Yale. And I'd like to thank the TissueGnostics group for allowing me to present my data.
Wow.
So thank you.
Thank you for that beautiful presentation. I hope you can hear me.
Yes, I can. Thank you.
Okay. Really, really interesting. It's so amazing to see you use the software for this. It's what it's intended for. So, wow, thank you. So I just wanted to open up the question and answer section now. If there are any questions, please write them in the chat. And yeah, in the meantime, while we're waiting, I just wanted to ask why did you choose 50 microns as the proximity cutoff between cell-to-cell interactions?
So when we were looking at, so what I showed you for the example were, so in our breast cancer cells, they have a wide range of morphology. And they can range from very small in terms of their size, so anything from like 20 microns to anything to about 50 to 70 microns. And so we felt that 50 microns was a good distance for some of our, not only for the smaller cell types, which is what actually I showed you here, but also for the larger cell types, if we were trying to capture things that are really in close proximity with each other. You know, and have the ability, likely the ability to have that cell-cell contact that we're really looking for, teasing out. That's like a really good starting distance. But we do plan, of course, you know, to actually look at other distances as well, so things a little bit closer, as well as because as I showed you just from those 50 samples, you know, most of the samples, most of our data was within less than 25, you know, microns, right?
Right.
And so, you know, even looking at data within a little bit closer range, maybe a little bit more useful as well.
Right. Okay. And so moving forward into the future, would that be, I guess that would be one of the ways you would expand the way you're exploring this data, right?
Right. And so one of the ways, so that's actually one way to expand it, is to actually use it as a sort of a guiding post. So one of the things that this actually brought up, and so in addition to, as I was saying, the really highlighting the fact that, there's still an appreciable amount of things going on within the tumor bed. This also allowed us, especially as looking at the proximity tool, where you're looking at, you're able to segment things into regions, actually gives you a lot of leeway in terms of like really separating things. In this case, I showed you in the stromal area, but if there are certain structures or certain architectures that are unique to the sample that you have, or unique to certain types of samples, it allows you to, really dive into that in a way that I think that, we may not have had in the past. So, so instead of just doing a one size fits all for the distance measures, you can actually make your own. And I think that that is something that's attractive, in order to follow up on, on a larger scale.
Totally. Wow. All right. Well, thank you. I don't see any questions right now from the chat. If nothing else comes in, then I have one last tiny question. They had a really beautiful an abstract image on slide 8. Was that a painting, or was that?
Yes, it was sort of like a sort of abstract, yeah.
I like that. Yeah, that was cool. Well, great. Thank you so much, Dr. Blenman. That was it was a pleasure as usual. And I look forward to watching this video again.
So thank you for allowing me to present my data. And it's a great tool that I appreciate having the opportunity to work on.
Of course. All right. Well then,
Thank you.
I'll let the team from EACR take it from here.